Good morning, everyone. Thanks for coming this early hour. So today I'll tell you about neural codes for 3D goal-directed navigation in the hippocampal formation of bats. This is the title of my talk, and there will be, I apologize for those of you who heard me last week, with some overlap, but not entirely, maybe 50%. And this part, kind of natural neuroscience, is more of a wishful thinking. We're not there yet, but this is just to make the point that I really believe that to understand the brain, we need to study it under conditions close to the natural behavior situations for which it evolved to function. And we're not there yet, but we're moving in that direction. And you can think of some of the things I'll be telling you about as an evolving story of us going more and more in that direction. I'll be more specific later on. So Yoram's talk was a great introduction, so I can skip some of the introductory slide. Our goal to understand the, generally I'm interested in the neural basis of behavior, but more specifically neural basis of spatial behaviors, spatial cognition, spatial memory, and navigation. And we study the hippocampal formation, the hippocampus of a rat here in this case, this purple sausage in the medial entorhinal cortex. And you've heard now, and you probably all know about the various spatial cell types that were discovered in the hippocampal formation. So in the hippocampus, you have place cells. So at the same kind, if an animal is running in a, let's say, about 1 by 1 meter box, then the black is the trajectory, and these are spikes, and this neuron will fire primarily in the northeast corner in this case. So this will be the place field of that neuron. So these are place cells discovered by John O'Keefe, and the grid cells we just heard about in the medial entorhinal cortex. There are also head direction cells, neurons that don't care so much about where the animal is in space, but more about the direction of its head. So you can think of place cells as a map, perhaps, a way to encode position. Grid cells could be used both to encode positions and distances, and head direction cells more of a compass. So you have basically all the ingredients you want: a map, distance meter, and a compass. All the ingredients you want to build a navigation system in the brain. There are also border cells, neurons that tell us the geometric borders of the environments. All this was discovered in rats, and we study this in bats. Why bats, I'll actually justify later on. I think it's easier first to talk about what actually are we doing. And this is kind of the question that will frame my talk, this referring to this natural neuroscience statement. So how does real-life navigation differ from navigating in a 1 by 1 meter empty box? That's kind of a bit of a provocative way to phrase the issue, but I think that's basically the way I'm thinking about it. And you can think of various ways, but just listed a few ways. So first of all, the world is not flat. It is complex and three-dimensional. And we've done a lot of work in recent years about characterizing the representation of two-dimensional space in the brain. I'll tell you about this today. Also, the world is not empty. It contains goals. And this is, I think, a fundamental issue, because if I want to navigate from here to the airport, which I'm going to do in the afternoon, so I apologize for the afternoon speakers. I miss your talk. I need to know, where is the airport? And from all what we heard right now, there is nothing about the airport in the representation. There is about where I am and about where is north, but where is the airport here? There's nothing about the goal, and I think we need some sort of a representation of goals in this system, and we have some new and exciting results about that, which I'll tell you about later. And also, of course, the world is not 1 by 1 meter in size. The issue of navigation, even in rats, occurs over kilometers, and so how does that work? How could this differ from the 1 by 1 meter box case? We are not there yet, but we are working towards that direction. I'll mention this at the very end. So these are the bats that we are working on, the Egyptian fruit bats, very large animals, at least by bat standards, 150 to 180 grams, means they can carry a lot of weight. So you can put on them neural telemetry systems, GPS data loggers, all sorts of equipment. They also have excellent vision. Just by looking at their eyes, you'll believe me that they see very well. So blind as a bat is a misnomer. Some years ago, we've done studies to ask how they navigate, like how does real-world navigation looks in these animals. And in collaboration with Ran Natan from the Hebrew University, we've put these GPS data loggers So these just store the position of the animal on board a flash memory chip, and you can glue it to a bat and release the bat. This is a wild animal caught in a cave halfway from between here and the Weizmann Institute. And this is an example of one animal that flies out of its cave to a local tree and then flies extremely straight to these two trees, feeds there for the entire night. These are fruit bats, so they're flying to fruit trees, feeds there for the entire night, and then flies back. And you note the scale here, this 5 kilometers, flies back extremely straight back to the cave. So these guys definitely can navigate, and they don't do it just for one night. They can do it for multiple nights. So this is now with an even smaller GPS data logger that was developed here by an Israeli company. This is an example of one animal flying from a cave to a tree 7 nights consecutively. So each color is a different night. And you see it's really flying to the same individual tree. Or you can't see. If you zoom in at the end, you'll see it's the same individual tree night after night. So they really can navigate, and we've done some translocation experiments to show that vision is actually important for this behavior. If they cannot see where the landmarks, then they are lost. So this kind of a very short introduction to what it means, what natural navigation actually means. And so this is what I'll talk about today. I'll tell you about 3D spatial codes in the bat brain, 3D place cells, 3D grid cells, 3D border cells, and 3D head direction cells. Some of it is published, some is ongoing work. Then I'll tell you about another unpublished story about orthogonal hippocampal maps for different sensory modalities in flying bats. Then I'll tell you about goal-directed navigation using vectorial representations. And then I'll tell you some future plans, which get to become more and more current plans as time goes by, but we're still not exactly there. So I'll start with 3D spatial codes in the bat brain. 3D place cells, 3D grid cells, and 3D head direction cells. These are studies by Michael Yartsev, Gili Ginosar, and Arseny Fickelstein. So to start with, I'll just I'm showing you this slide that summarizes several years of work. But basically, the take-home message here is that in 2 dimensions, we found all the same spatial cell types as in the rat. So in the rat, there are place cells, grid cells, border cells, and head direction cells, and we found the same kinds of cells in bats, and they are very similar in many of the detailed functional properties that we characterized. There was one main difference in that the oscillations that are characteristic of hippocampus of rats were not there, or were very different in the bat. And I will not talk about this at all today. So those of you who want to hear about this, come talk to me in the coffee break. But in terms of the functional properties of the neurons, they're generally very similar. Then you can ask, okay, this was in 2 dimensions. Now, how about 3 dimensions? So, if this is your 2-dimensional place field, so you could imagine that in 3 dimensions you'll have, a spherical firing field, or maybe not. Maybe it'll be a cylinder, so there'll be no encoding of height. And so, to test this, we worked for several years with Michael Yartsev to develop a telemetry system that allows recording spikes in flight. Okay, so it's wirelessly transmitting the data. And so here you see an example of recording from the 4 channels of one tetrad, and you can see here the spikes. This is recorded in a flying bat. And we also had to develop a behavioral test, how to convince a bat to fly around and around and fill the volume of space, which is what we want to characterize things, which turns out to be tricky, because if you give them a very small number of targets, they'll just develop very stereotypic kind of habitual trajectories. But we've used this kind of artificial tree, which has 8 arms, and randomly put food in there, and then the animal really fills up this nicely fills up the space that we have, which is a flight room of about almost 6 by 5 meters. We could reconstruct the 3-dimensional position of the animal using 2 cameras. And then we combine the behavior and the spikes. We get something that looks like that. It's an example cell that indeed was active within a spherical region of space, meaning whenever the animal flew through more or less a sphere, the neuron fired. So this is kind of the spherical-like place field of this neuron. Indeed, in more than 90% of the neurons, they were not significantly different from spheres. So they were really spherical in shape, meaning that the resolution, the x, y, and z dimension were the same. So we kind of concluded that there is an isotropic representation, meaning the resolution is the same in all directions. Also, in this particular condition, the representation was uniform, meaning place fields tiled space uniformly, although in other conditions, experiments that we are running now, where you have very important positions in space, like landing platforms, then there is kind of clustering of place fields around them. So it's the uniformity is not a general statement, but isotropicity seems to hold so far as far as we can tell. Okay, next you can ask, how about grid cells? And this is an ongoing project. We're still trying to understand what's going on, but this is basically the question. So if in 2 dimensions, this is how grid cells look, kind of an optimal packing in 2 dimensions, in 3 dimensions will look something like that, or will you have vertical columns or something different? And so Gili Ginosar has recorded a number of neurons. She's actually running right now. I think she should be here, but she's running these days another bat, and she found some neurons that look like that. Okay, so this is a neuron that, first of all, it looks very different than the neuron I showed you before in the hippocampus. Okay, it doesn't have this one activity blob in space. It has these multiple blobs. But it's clear that just by looking at that, it's difficult to tell what's going on. The looks seem to be some periodicity, some structure to it, but you need to find ways to characterize that. I can already tell you, and I realize I didn't put a slide in here, but Gili has looked at distances between, so local distances between the blobs. So you can look at, distance between that, activity field and its nearest neighbors, and then that one, its nearest neighbors, etc. And look at the histogram of these distances, and the histogram turns out to be very narrow, meaning that the local distances between blobs are much more reproducible than in a random set of firing fields. So it seems to be that there is a local scale in the activity pattern, which would be useful if you want to encode local distances. But the question is there also a global scale? Is the global lattice? Because that will be crucial for something like the grid code that Yoram described. So the question is there just a local scale, which we already see that there is, and I don't have the slide here, versus a global one? This will be the key question that we'll look at. And so to look at the global structure, we are still struggling with ways to look at that. But the first thing that Gili did is to look at symmetry properties of grids. So for example, if we assume that there is an FCC grid, which is one of these optimal sphere packing grids, then an FCC grid is based on a tetrahedron. So you have like 4 planes of symmetry through which you can cut the perfect grid, and you'll get this 2-dimensional hexagonality. So this will be one plane that is the second plane, third plane, and fourth plane at the back. So 4 symmetry planes. And so the approach here will be to take the data. This is just a simulation, of course. Take the data and take a plane, cut a plane through it, and rotate it through various angles of azimuth and elevation. This is the approach that we borrowed from Alessandro Treves, who I just noticed sits back there. And what Gili found is the following. First of all, if you look at simulated data, so this is the perfect FCC. So if you plot the elevation versus the azimuth of the plane, and the color code is how much hexagonality, how much 2-dimensional hexagonality is there in that plane. You see, for a perfect FCC, you have these 4 hotspots, 4 high hexagonality blobs, which are exactly the 4 symmetry planes of an FCC. So this is what you expect. If you take columnar structure, then you have just one activity pattern. You can think of this, one blob. You can think of this as Greenland, and this is Antarctica. So it's really one plane with which you can cut these cucumbers, and you'll get hexagonal structure. Okay, so this all is straightforward. Now, for that particular neuron that I showed you before, this is a real neuron. This is what we get. It's not exactly like that. Still, these 3 blobs are in there. This kind of 4th blob wandered somewhere. You have some of the negative values. This seems similar. It looks similar-ish to that one. It's only very different than this, and very different than a random set of blobs, which I'm not showing you here. Those basically look like the center of this, like static noise. So there seem to be that there are some cells that have also a large-scale structure, a lot of cells that have a local structure, yet other cells, and this is a zoo, so I'm kind of, I don't know if you've noticed, I'm in the process of describing all sorts of animals in a zoo. So this is yet another kind of animal that we see, and we have several of those. Neurons that have elongated stripy structure in space, so seem to have a preferred axis. And there are also quite a few cells that look like border cells, that will be three-dimensional border cells, will be active along the floor of the environment or along the wall of the environment. And some cells, we had even a few cells that seemed like almost like the border cells that are active on a few walls of the environment, but on the wall, the activity is blobby. So it's almost like a border by grid cell or something like that. So it's really a zoo out there, and we are still, I mean, that's where we stand. We're trying to find more ways. We're now running, we have a visitor student from MIT who's here also, Robbie, who's running now Fourier-based methods, basically inspired by crystallography. So we're trying to figure out, is there also a large-scale structure, or is it just a local scale? Okay, what about head erection cells? So I mentioned already that in rats and in bats as well, we have azimuthal, horizontal head erection tuning. What about three-dimensional compasses? Do we have neurons that encode, you know, that direction versus this direction versus that direction? So this was a project that was run by Arseny Finkelstein, a PhD student in the lab, and Dori Derdukman when he was a postdoc in the lab, now is a faculty at the Technion. And so what Arseny and Dori did was to develop this device, kind of a non-coplanar array of LEDs that I'll spare you the details, but basically allows you to unambiguously measure all the three Euler angles of the head, the yaw, the pitch, and the roll in a behaving animal. And then we can record from the presubiculum in the bat, which sits in between the hippocampus here and the internal cortex at the back. The presubiculum is the first, the dorsal presubiculum is the first place in the brain where head erection cells were discovered in rats many years ago. And then Arseny found the following cells. So here you see examples of three neurons. So this is a pure, so this is, what you see here is the pitch versus azimuth, and the color is the firing rate. So you see that this neuron doesn't care so much about pitch, but is encoding azimuth. So this is your classical rat-like azimuthal cell. But Arseny also found neurons that look like that. They don't care so much about azimuth, but care about pitch. So this neuron cares about direction down. And you'll note, by the way, that the range here is limited to plus minus 45 degrees because this experiment was done in crawling bats. Okay, so then crawling belly down, you know, the head goes up and down about 45 degrees. Normally, they can't do this. So I'll talk about experiments when they actually do the full 360 degree of pitch later on. And Arseny also found neurons that encode combinations, like this neuron, a combination of pitch and azimuth, and there are even a rare, not very common, but a few neurons that encoded all three Euler angles, so pitch by azimuth, and roll by pitch. So overall, of the azimuth-tuned cells, about 35% were conjunctive, primarily conjunctive to pitch. Roll tuning was generally pretty weak. Most neurons were azimuth by pitch, meaning encoding a real direction in space. So a neuron that likes that direction or this direction or that direction. So there are three-dimensional compasses in the brain. Next we asked, and here came the surprise, we asked what happens if the animal is inverted. And of course bats like to be inverted a lot of the time. And when we ran actually two sessions, one upright, another upright session an hour later, but in between there was an inverted session. And here you see examples of three neurons. And you see that this neuron is tuned to a particular azimuth. This is azimuthal tuning. And there's the same azimuth an hour later. But in between, it's still tuned, but to a different angle. And the same for the second cell and the same for the third cell. They tuned in the inverted position just to a different angle. And if you stare at it for a few seconds, it took us actually 3 months to figure that out. You'll note that this, it's not just random shift, but actually shifted by 180 degrees. If you shift those tunings by 180 degrees, then all these three neurons align, and not just these three example neurons, but in fact across a population. If you compare the preferred tuning in the upright session, the first versus the second upright session, then the difference in preferred angle is around 0 across the population. This just means stability of the compass. But if you compare the upright versus the inverted, then the difference is around plus minus 180 degrees. So there's inversion across the population. And we interpret this as the head erection signal is encoded in toroidal coordinates. So on a torus, you have 360 degrees of azimuth by 360 degrees of pitch. So in a toroidal coordinate, this and this and this and this and that position are all the same azimuth, which means there is actually no inversion of 180 degrees if you assume this toroidal representation. And why would something like that be useful? Because a torus is continuous, whereas a spherical coordinate system, which is probably the first thing you think about when you think about 3D angles, has a problem of singularity at the zenith, right? If I'm a head direction cell and I like that direction, and I'm now starting to pitch, and I'm pitching up to 89.9 degrees, I'm still firing, and then I pitch, you know, an epsilon further to 90.1 degrees, and then I'm supposed to stop firing abruptly. So this is, first of all, non-biological, presumably, but also for an animal that's doing all sorts of crazy maneuvers in 3D, it just doesn't seem desirable. So this has the property of continuity. And indeed, if we compare the correlation between the firing in upright and the inverted condition, the data are much more consistent with a toroidal than with a spherical coordinate system. And Arsenio also did some tests to explicitly test some predictions from this model, so including having bats crawl in a box where they either crawl on the floor or could do a full 360-degree maneuver. So basically there's food on the floor and on this ring, so basically a mesh ring, so they would crawl a bit, then do a ring, crawl a bit, do a ring, just volitionally would choose what to do. And what Arsenio found that there are neurons, basically all the neurons that were tuned to pitch were tuned unimodally. Okay, they were tuned to one particular angle that could be anywhere across the range of 360 degrees of pitch, which is predicted from the model. And also, you can now compare what happens on the floor and on the ring. So this is an example of a neuron. This is the tuning to azimuth on the floor. This is the tuning to pitch on the ring. And now you can just multiply the two, build a model. Of course, now we have here in the model 360 degrees of pitch, 360 degrees of azimuth. We not actually experimentally sampled all this space, right? This is just multiplying these two one-dimensional curves, assuming independence or separability. But now we can compare it to the real data that was recorded on the floor in this plus minus 45 degrees range that I mentioned to you. And you see that the real data is very similar to a cut through plus minus 45 degrees to the model, which is, again, suggesting that this is consistent with the torus, and it's not just for this neuron. If you do the correlation coefficient of that, the real data, and this piece in the predicted model, then the correlation coefficient across the neurons are pretty high. So it's a general result. And if you connect that part to that part and this part to this part, you'll close them together, you'll get a torus. So this is how it looks in actual toroidal coordinates. And Arsenio also did experiments in flying bats and found essentially the same results. You have pure azimuth cells, pure pitch cells, and azimuth by pitch cells. So conjunctive neurons representing a particular direction space. The main difference was the firing rate was much higher in flight than crawling, which is just something we generally find in all these types of spatial cells. So just to illustrate for you why this will be important for a flying animal like bat to have a toroidal representation, I'm showing you a movie. This is a slow-down movie of bats landing on a grid on the ceiling. Well, grid is a bad word, on a mesh on the ceiling. Okay, and you see that it goes through the zenith, or the plus 90-degree pitch right there. It's slowed down 10 times. In reality, it happens 10 times faster. But this is just to illustrate that this issue of toroidal versus spherical coordinate frames is just not just a contrived mathematical question. It's really very relevant for an animal that's doing this all the time. So just we'll see a few more landings and how the animal goes to the 90-degree pitch. And of course, when they take off, they go to the minus 90 degrees. So the same problem is there. Okay. So the features of the toroidal representation, I already mentioned the continuity. And another interesting feature is the commutativity. In general, 3D angles are non-commutative, meaning the order of how you do the angles is important, which creates all sorts of complications. You need to store the memory of the order of things you did in the past. It's really, it becomes a very complicated problem. And we think that the bat head direction system can solve this problem in a very neat way. It combines a neural trick and a behavioral trick. The behavioral trick is do not roll. And in fact, a lot of animals avoid roll, like this bike rider, or a lot of animals seem to avoid roll, and it's usually explained from a sensory perspective. Why do animals not roll? To stabilize visual input. We have a different explanation for this, because if behaviorally you don't roll, which bats don't do much, and neurally you represent your head direction on a torus, then the angles become commutative, and this simplifies everything. So this is another nice feature of the toroidal representation. And of course, we don't know. This is all in bats. what of these things apply to other animals. Of course, many animals move through three-dimensional space, like monkeys or dolphins or rodents that climb very well, or even humans. I'm looking down from a 8th floor versus at ground level. I know something about my altitude or direction in space. So I think it's a general question that's interesting about three-dimensional representations across species. Mice, for example, like to hang upside down in their cages. Anybody who worked here as mice know I've seen them do that. So it'd be interesting if they have toruses in the brain as well. The next story that I'll tell you about is a study by Maya Geva-Sagiv, who is actually a student at the LSEC ICNC program. It works with me. And this is about orthogonal hippocampal maps for different sensory modalities in flying bats. So the basic question that, Maya asked is what happens if you compare, if an animal maps space using two different sensory modalities. So bats have two good senses. They have good vision, as I already mentioned, but they also have a sense of echolocation. They can use sound to map out space. And we've done some studies about echolocation per se. I won't talk about those, but you know the basics. They emit a sound, echoes come back, and they can reconstruct the distances and directions and even identities of objects through that in complete darkness. So what Maya did is the following experiment. She trained bats to fly kind of on we call it a linear flight track. So just from one side to another between two landing balls. And they can do it either in complete darkness, 1 microlux light level, so pitch dark, and then they rely on echolocation, or they are flying in the light, but then Maya plays these broadband sounds that mask echolocation. And if you do both, you turn off the light and broadcast the sound, then the bats crash. They cannot do this, so it works. And also we put ventilators to blow away any odor trails in midair if they do exist. So the idea here is to try to create as much as possible a dissociation between these two conditions, a sensory dissociation, pure echolocation versus pure vision condition. And then she records neurons from the hippocampus. And these are some examples of cells that she found. So let's focus on this neuron first. Is all from one neuron. These are just two views. The XY is top view, XZ is side view. And this is kind of the X is the direction of the kind of the long axis of the flight. And you see that, and we have here 3 sessions: a vision, pure vision session, then an echolocation session, then a repeat of another vision session. And you see that this neuron has a place field on the right side of the room in the vision, and again in the second vision session, but literally 0 spikes in echolocation. And this neuron is doing almost the opposite. It has a place field in echolocation, but doesn't fire almost at all in vision. And then you have a cell like that has a place field on one side for vision, another side for echolocation. It kind of shifts around. This one shifts around. This one shifts around a little bit. And there are some cells that seem not to shift at all. So we kind of have a range of differences and similarities between the 2 sessions. Now, it turns out that these kinds of neurons that are kind of the on-off ones that like one sense or the other are concentrated primarily in the subiculum, because Maya actually recorded in 2 subregions of the hippocampus, CA1 and the subiculum. Subiculum is kind of the white spot, terra incognita of the hippocampus. We know really very little about why do you need a subiculum at all. It's a major output station of the hippocampus, and very little study. Even in rats, there are really very few studies comparably to CA1 certainly that were done. And what Maya found is that in subiculum, 60% of the cell did this kind of thing. So very dramatic kind of remapping, what we call. Whereas in CA1, where typically place cells are studied, many less neurons did that. In CA1, something else happened, and the neurons did this shift kind of thing. They shifted their place field around. And you see that we have this remapping index, which is 1 minus the correlation coefficient between light and dark. And you see that this remapping index is much larger for CA1 for subiculum, just indicating that they tend indeed in CA1 to shift their place fields around. And now we can do the crucial comparison. We can ask, okay, so we have this histogram of these remapping indices for the real data in blue. And now we can ask, is that random or not? So we can now take and compare all possible pairs of place fields in the dark and in the light of non-identical neurons, okay? So this will give you a kind of distribution across the population, the null distribution across the population, if you will. And this, so a cell shuffling kind of approach, and this gives you the black histogram, which is visually identical to the blue histogram and indeed statistically non-significantly different. But the point of that then is that really the remapping, the shifts in place field that you see between the light and the dark are completely random, meaning the maps in the light and the dark are statistically orthogonal. And this, to us, suggests that really the idea that you have a unitary map, an abstract map in the hippocampus doesn't seem to be the case, at least in our conditions, right? So if the idea, John O'Keefe's original idea was place cells are representing the position of the animal no matter what, then it should be abstract regardless of whether it's light and dark. And I just showed you that the neurons actually are really orthogonal between these two conditions. So, we suggest this, instead of a cognitive map, a cognitive atlas hypothesis. So the idea is just like in an atlas of a certain country, say an atlas of Israel, you have, you have a road map, you have a geological map, you could have a political, political map is better to talk about here. No political maps, okay, whatever. Some other map, and so these are all maps of the same space, right? But they're used for different purposes. And so similarly, under different conditions, light and dark, for example, when obviously, we think of ourselves navigating somewhere, then, you use completely different landmarks to navigate under these two different, night versus the day. So, so you'll have, two different pages of the atlas. So for the same space, and somehow you need later on to align them, but this doesn't seem to happen in the hippocampus. So this will be kind of our take on these results. Okay, and finally, how much time do I have? I have time, okay. So finally, this last part of the talk is about goal-directed navigation using vectorial representations. This is the work by Eilert Sarel, and again, Arseny Finkelstein. And so to start off, these are experiments that were run by Yossi Yovel and Ivo Borisov and myself, asking basically how is now the long-term structure of the behavior of bats in the field. So for that, we had an Israeli company develop for us an even smaller GPS data logger that can now record data for several months. And so this is an animal where we recorded 75 nights of data in the field. And I'm just going to show you some of these nights. But the animal in this case left the cave and flew to a particular tree for several nights. Actually did it much more than I'm showing you. Did it for 2 or 3 weeks. I'm just showing you a few nights. It becomes cluttered if you look at more. But and then it switched to another tree. Of course, at the end, the fruits are depleted. They cannot fly to the same fruit around the year, right? It's obvious. So they have to switch to another goal, another tree. And in some days, like day number 33 here in Cyan, the animal started flying in this direction, then changed its mind and did a shortcut to this other tree. And you also had some other cases the animal did this kind of thing or did this, and then to yet a third tree that's around here. So they do all sorts of interesting behaviors. But the point of just showing you that is that they definitely seem to know where the trees are and fly very in a goal-directed manner. So, where is the representation of goals in the brain? And so now, people thought about this question, and typically in the hippocampal field, the kind of typical explanation is that you can use place cells to do this encoding. So if I want to plan how I'm going to walk from here to these nice flowers over there, I can sequentially activate place cells that represent this trajectory. So this is called pre-play, and I can use that to plan this trajectory. Now, if you look at that, you immediately see the problem here. So first of all, the scale, if you scale up now to 100 kilometers, then you'll need to activate a lot of neurons. It seems to become more complex, and even worse, this pre-play that people have found in the hippocampus has a kind of a fixed speed in meters per second. So if you want to pre-play this entire distance, the bat will presumably need to sit here for 10 minutes just pre-playing this, at least if we extrapolate from laboratory studies. So it seems to be that there's a problem with this kind of thinking. So we came up with a different idea, and that is all you need to know to represent the direction to this tree is to know the direction, right? All I need to know is this is where the tree is, the angle. Okay, the nice thing about angles is that they're invariant to scale. 360 degrees is 360 degrees, no matter if you do it on a 100-kilometer scale or in a room. It seems to be a much more compact way of representing things. So Ailton and Arseny indeed went on to study this in our flight room. And so they have bats flying around in the room, and there's a goal. There's a platform that looks like that, and the bat flies around and lands on the platform. And so then we can ask, okay, if this is the heading direction of the bat, okay, this is where it's flying. We define this as the goal direction, okay, the difference between where it's flying and the instantaneous direction to the goal. And then you can ask, so this would be a goal direction of 0, and are there neurons that preferentially represent goal direction 0, preferentially represent the direction to the goal? And surprisingly enough, at least for, I think for most hippocampal people, they did find such neurons in C1 of the area of the hippocampus, which has been studied for 40 years. So neurons that are tuned to angle 0, to goal direction 0. And I just want to stress again, this goal direction is not a compass, right? If Yoram is my goal, so now this is goal direction 0, but if I'm moving over here, okay, so this will be goal direction 0, okay? So it's not an absolute compass in space, it's really relative to the goal. So it's different than the head direction representation that I showed you before. So these 3 neurons are tuned to goal direction 0. They also, Ayelton Arseni also found neurons tuned to other angles, other goal directions, and if you look across the population, it looks something like that. You have, you have all possible goal directions across the population. It's all preliminary data. Okay, so take all the histograms, this histogram with a grain of salt, but there's overrepresentation of cells that like this direction, so goal ahead if you like, or and 180 degrees, so goal behind. So, but anyway, there are all possible angles here. And this is not just somehow resulting from the spatial tuning that we know exists in the hippocampus, because now we can look at the spatial tuning of, let's say, of this neuron and ask how does the goal tuning looks if we take the data around the place field versus the little firing that we have outside of the place field. And you can compare the two, and they're not exactly the same, but they're kind of similar. And then you can look at the same for this neuron, compare the infield and outfield goal direction tuning, and this neuron looks really similar, and that neuron looks really similar, and this neuron, it also looks kind of similar. You can compare now across all neurons, and we find that the difference in goal direction in and out of the place field is around 0. It's not really as, whenever you go through a place field, you have a particular preference, but it seems to be across space. And finally, goal direction tuning is stable across time. So you can now ask, every time they land, you can create these tunings that are now color-coded, at time 0, at time -4 seconds before landing, -8 seconds before landing, and they're flying fast. So -8 seconds is many meters before the landing, and the idea is this is stable. And again, it's not just these 3 neurons, it's a lot of neurons that do this. So it's not just only at the very end when they approach a target that they have this tuning, but they have this stable tuning throughout. So to summarize these preliminary results, we found a subpopulation of hippocampal neurons that exhibited rather narrow angular tuning to goal direction, and this goal direction signal suggests that there is a vectorial representation of goals in the bat hippocampus, which could support goal-directed navigation. Okay, so where are we heading? A few words about future plans. We're starting to work on optogenetics. This is also done by Gili Ginosar, and so far we see that viruses at least are able to infect the neurons, so we're working on this right now. And another direction is asking this large-scale spatial representation, this question asked in the beginning, right? That this is real navigation for bats, but also for rats that can go for a distance of 1 or 2 kilometers per night. And we really don't know if what we found in small boxes translate to this or not. Just to explain why this is a non-trivial question, if you look at this cartoon and you think, okay, if I need to tile a distance, of 20 kilometers, and the flyway that I showed you is about 2 kilometers wide and 500 meter high, if you want to tile that with place fields that are 10 centimeter in size, you'll end up with 10 to the 15 neurons. If you assume 1 meter size is 10 to the 12 neurons. Either way, it's orders and orders of magnitude more than the million neurons or so in the hippocampus. So it just cannot work. So there must be something else. So what are the possibilities? For example, it could be that there is a hierarchy of resolutions. For example, it could be that important places like the cave and the tree, you'll have high resolution, small place fields, but high up, you'll have, in the middle of the flyway, you'll have 100 meter or 300 meter size place field. So why does the animal need to know its position in the middle of the flyway to a 10 centimeter accuracy? This also behaviorally doesn't seem necessary. And then you can solve this issue of number of neurons This seems plausible, but was never tested. Another option suggested by Ketch and Zhang was that instead of having these very large fields, it's better to have multiple place fields. So they have a place cell, instead of having one receptive field, has hundreds of receptive fields. This is optimal from a decoding perspective. This is unpublished work by Ketch and Zhang. This is a completely different suggestion now. So all of this is really unknown. There could be, of course, something completely different on large scales. So to address these questions, we've developed this neurologger, a device that allows to record data on board the animal. Now it's different from the telemetry system that I showed you before, which is transmitting the data. So it's great for indoors, but is limited in range. To record over unlimited range, you need to record the data on board the animal. So this is what I'm showing you here, and you can record nice spikes with that. And so we are really, I think, very close to starting answering this question. Okay, so let me summarize. This is my last kind of summary slide. Why do we use bats? So first of all, and really I didn't mention this almost at all in today's talk, they have this temporally discrete sensory input, this sense of echolocation, which is very interesting in its own right. But the nice thing about it, you can put a microphone and record with a 1 millisecond precision the timing of sensory input, which turns out to affect the firing of place cells. I didn't show you that, but it's a work that I've published in the past. And in a rat or a mouse that runs in a maze, it's just a difficult measurement problem. How do you measure when exactly did it see something, touch something, smell something? It's much more difficult. In a bat, it's relatively easy. Just record the echolocation. So that's a nice advantage. You can study 3D. Theta oscillation, which, as I said, I didn't talk about at all today, but it's very different between bats and also humans and monkeys on one hand, and rodents and rabbits on the other hand, which narrows the range of possible computational theories of hippocampal function. They have excellent spatial memory. We would like to bridge the gap between behavior in the lab and in the field. And the two most important points for me, one is this comparative systems neuroscience. At least in the hippocampal field, there have been an enormous, fantastic amount of work, but it's all done in rats and a little bit in mice. There's compared to sensory systems that were studied across species, there's very little work on other species. And I think, at least as a sanity check, we need to see what works in all, what's invariant across species and what isn't. And the second is this issue of natural behaviors. I think, and in any system, be it sensory systems, but certainly in a navigation system, we need to get closer to what natural navigation looks like, and in order to understand brain function. So I'd like to thank lab members who bravely explore 3D space. This is from a lab trip that we did in September. Here we explored vertical space primarily. And here you see Arsenia and Alon checking what's going on when you pitch your head above 90 degrees, and Tamir pushing the limits of 3D in humans, at least, in bizarre ways. And these are the lab members. I'd like to especially thank Lior Alas, who is my right hand in the lab, and many of you know her, and she's fantastic. And the lab members, I mentioned most of them as we went along, and alumni and collaborators and funding sources. And thank you for listening.